Okay, so I guess it's time to start this session, understanding the mechanisms of nano interacting with life. And our first speaker is Prof. Barbara Rutishauser, please. Oh, you are switching. It's Prof. Alke Fink, so please. They are sharing the first talk, which is unusual.
Thank you very much for the invitation, for the introduction, and all of you that you're here. My name is Alke Fink, and together with my colleague, Barbara Rothen, we are responsible for the BioNanomaterials group at the Merkle Institute in Fribourg, and also together we are giving this presentation, which is on nanoparticles and how to sort of behave or how they are characterized in complex environments. And I think most of us, or all of us, have seen graphs like that show actually the big difference between what is published every year. So this is the solid line here in terms of particular materials and how they are to be used in nanomedicine, and the number of materials, the same or respective materials that are going into the clinical phases, right? And this review here, they mention, let's say, the more traditional nanomaterials based on polymeric formulations or on liposomes. But if you're looking at inorganic particles, the trend is obviously the same. This is interesting in a way because if you look at antibodies that have, give or take, like the same size, if you think in terms of nanometers and diameter as these materials do, and there the behavior is different. So you will have much more in clinical phases than with these original or these, let's say, more material type approaches. So now the question is, why is that so? So why are the clinical trials lacking behind all the research that is out there? And I also guess that many of you know this very famous paper by Harald Krug. He looked at this or tried to find explanations not for nanomedicine, but for nanotox. But I would say many of the points that are raised in this work can one-to-one be translated to nanomedicine. So it's about particle characterization, it's about assay development, assay validation, the right number or amount of controls, interferences, the comparisons between the labs, and something I want to talk about today is the particle characterization in biological environments. This, of course, is not the answer of why this is lacking behind. There are so many biological or physiological reasons too. This is really from, let's say, more materials perspective. So what I did here is I placed a gold particle, it's a 40 nanometer gold nanoparticle, in an environment it would encounter in a simple cell culture assay, right? So this is DMEM in 10% serum, and you see here this particle is not alone. There's a lot of stuff sort of in very close proximity. You see some of it approaches the size of the particle itself. These are obviously proteins. You have lots of smaller molecules, and you see this green sort of tint of the entire box. These are electrolytes, which is something that particles or any colloidal system is not particularly fond of. So if you imagine a particle in such a, let's say, busy or crowded environment, what can happen to the particle? The number one thing we've been talking about for many, many years is protein corona. So the interaction of proteins that are sort of swimming in the same soup as the particle, that they interact with the particle surface, and they form like a new identity, if you will, of the particles. Another thing that can happen, not to all the particles, but to some of them, is oxidative dissolution. So you can actually dissolve your particles. Another thing is particularly frustrating if you're a chemist and you manage to put a lot of drug molecules, antibodies, what have you, to the surface of the particle to achieve any sort of targeting, you can just lose this entire payload, if you will. And something the community has started to discuss since a couple of years is obviously aggregation. Since this is packed with proteins, with electrolytes, so many of the particles might indeed start to aggregate. So now, how can I characterize nanoparticles that are sooner or later to be used in an in vitro experiment, in an in vivo experiment, in a human being? And this environment, I think we all, just as from a chemical perspective, is very different to what we do in like buffer or any aqueous solution where the particles are prepared in. So these are all what I call complex media, cosmetics, foodstuffs. Well, this is blood. I'm focusing on, let's say, more the biological fluids in this talk here. So why do we need so many different machines to characterize particles? In German, we have a word for that. This is called the eierlegende Wollmilchsau. This is pretty hard to translate, but at the end of the day, what you would want to have is one thing to answer all questions, like here, this animal that can, you know, produce eggs and milk and wool, and, well, finally, you can probably even eat it. So you might want to have something that will help you with hydrodynamic size, core size, magnetic size, charge mobility, blah, 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 blah. So there is much more. Obviously, such type of machine does not exist for the simple reasons that all the instruments, they make use of a particular physical or physicochemical property of the material you want to characterize, and this sort of defines a little bit like the technique. So why is it hard to characterize in this particular environments? Because you have this large amount of proteins, and these proteins end up having pretty much the same size as the particles, and this can be pretty confusing. And this is why this is something we did very recently. We used a standard DLS instrument, something that many of us have sitting in their labs. We measured gold particles, so this is the green distribution, but we also measured without any particles, just a cell culture medium with a serum, and I think we can agree that this can be pretty misleading, right? Because all you're measuring here is proteins. What you can do with some particles, again, not with all particles, because we don't have this eierlegende Wollmilchsau yet, you can measure in depolarized mode. So, and if you measure depolarized dynamic light scattering, you can sort of filter out the signal that comes from the biomolecules. This is something we've been doing here. We have different types of gold particles. Here you have them in a buffer. This is always the dotted line here. And then you put them in different environments. Like here, we keep them in a buffer at BSA. You can see how the particle increases its hydrodynamic size just because it absorbs the BSA on the surface. You can see that in cell culture medium, within 5 minutes, they strongly start aggregating. If you add serum components, they can be stabilized. Stuff like that actually works very nicely with some of the particles in this depolarized mode. What we've been starting to do in the past couple of, well, in the last year, we looked at so-called Taylor-Aris dispersion analysis using some sort of capillary electrophoresis setup. In essence, you have a liquid that contains the particle, you pump it through a narrow capillary, the liquid flows under laminar conditions. So in this laminar flow, this initial plug, so where you inject the particles, it is stretched out. You have a couple of detection windows, and essentially you measure again diffusion. This is something that the chemists or the biochemists have been used for many years to characterize proteins, and we try to see what can be done with particles. As always, we start with model particles, so these are monodisperse in size and in shape. So the usual suspects, which is silica, small, medium, large, gold, small, medium, large, and these are the magnetic particles. Here are some 10 micrographs, and more or less this is nicely comparable for most of the cases with the light scattering data. I'd say as an advantage to DLS can be said that with a Taylor dispersion analysis, you can actually access the really small particles as well, something which is very hard to do with light scattering. You can measure individual molecules like the albumin, so this seems to work very well. Now what happens if you go to more, let's say, industrially available particles? That means, you know, less nice and monodispersed as the ones I've showed before. We looked at zinc oxide, titania, or copper particles, something that is very, very difficult to reliably measure in DLS. This actually worked very, very well, but I cannot hide that we have to adapt the algorithm which we're using to fit the functions. Also this is no black box, you know, where you just input a particle and click a button. So you will have to look at the data really carefully. So now the question is, the numbers that we get from that make sense. So now what happens if I now go back again to my model particles? Again, the gold particles, pretty small ones with different surfaces. And again, like the first image I've showed you, I place them in these different environments, pure PBS, a PBS that we spike with some BSA, a typical representative of cell culture medium, and then there where we add the serum. All this is very reliably measured and easily compared to what we've done with DDLS. You can track the stability over time, which actually works really well. You can selectively detect. There's 2 detection windows. You can also, for example, detect a particle and a fluorescent dye that you add on a surface. So you can also figure out how pure your sample is, or if what you graphed on the surface gets desorbed. And the only thing we are currently worrying about is that some of the particle suspensions are very, very sticky, so we need to modify the capillary in some of the cases.
So after we did now all this very complex characterization of the nanoparticles into complex media, the next step is then to add the particles in a biological environment with cells, like here with macrophages, which are cultured, and you see that they immediately take up the nanoparticles over time. So we all know that the detection of the nanoparticles in cells or in tissue then needs somehow different methods, as we have just heard from Alke. And also the quantification of intracellular nanoparticles sometimes uses other methods. We can use chemical analysis, which is of course the same as particles in a buffer solution. But then for the intracellular quantification, we also need to get rid of the very complex bioenvironment which can induce artifacts during the measurements. And it is very important that we relate the intracellular nanoparticle concentration or those with a possible cell response, because this is important for the safety assessment and also for the regulation. So most of us usually need fluorescent particles to assess the distribution of the particles, the kinetics over time. And this is mainly based on detection methods for fluorescence. So we have actually worked quite some years to produce these gold nanoparticles with a fluorophore on the surface, because gold is biocompatible, it is inert, it's not degraded, so it's quite nice also for animal studies to study the kinetics, the distribution in the organs. And so what we had to do is we needed to shield here this attofluorophore on the surface in a polymer, in 2 polymer layers, first to have a big enough distance from the gold core to avoid quenching, but also to avoid that the dye directly interacts with the cell surface, because this can already change the interaction with cells. So after we worked out the synthesis for these particles, we have tested positively charged polymers and negatively charged polymers, added this to the cell cultures, and the first step was to detect the particles by confocal laser scanning microscopy. So for the positively charged particles, we saw quite some uptake here, the red dots in the cells. However, for the negatively charged particles, we didn't see any particles. So the first conclusion is negatively charged particles are not taken up of the cells because they have a negative surface charge of the membrane. No, this is not true. This concept does not work, because if we look with other methods, like with transmission electron microscopy, where we can see the gold core of the particles, you also see that for the negatively charged particles, They are inside the cells in the endolysosomal system, and they are quite numerous in comparison to the positively charged particles. So this, there is something different which we didn't know at this time. So we also did spectrometry methods to identify the gold by digestion of the cells, and we see an uptake over the first hours which is significantly increased after 24 hours, where we saw 60% of the particles which we gave into the system were taken up by the cells finally. So these pictures then started that we thought a little bit more in detail about this, and then we decided to investigate the stability and also the fluorescence in a more at lower pH, because these endo and especially the lysosomes, they have a low pH between 4.5 and 5 and a very high ionic strength. So we mimic this lysosomal environment by producing this artificial lysosomal fluid. They do not have the enzymes, but all the other conditions are quite similar. And what we observed was that in this artificial lysosomal fluid here by UV spectroscopy, that the particles agglomerated. You see a broadening of the spectra, and also by eye we saw an agglomeration of the particles, which we could not observe in PBS buffer, and we also did not see this for the positively charged particles. We did another experiment where we isolated the lysosomes after the cells have been incubated with the particles. And again, we have seen an agglomeration of these negatively charged particles in this acidic environment, but only the negatively charged particles. So by surface-enhanced Raman spectroscopy, we have seen that somehow the polymer was swelling, and this induced a decrease of the distance from the fluorophore to the gold core, which then finally resulted in a quenching. So it was just the environment which has reduced the fluorophore properties, or decreased these fluorescent properties, and we still have particle uptake. So we have to be very careful with all these investigations. So what we try now also with other systems, we try to assess the colloidal behavior and the long-term stability of any particles we produce in the lab with this artificial lysosomal fluid platform, because it's quite easy. You just incubate the particles, you incubate it for weeks or several months in the incubator, then you assess the stability or the size, the hydrodynamic diameter, or here just by transmission electron microscopy, you already see that silica particles in water and artificial lysosomal fluid after 1 month are still looking quite similar, whereas for iron oxide particles, we already see some change in comparison to the particles in PBS or in water. And after 3 months, we even see a more significant change of the particles. We need now, of course, to complete this experiment with a more characterful measurement of the particles. So with this, I would like to already conclude. So if we, I hope that we could show that nanoparticles in a biological environment need very complex analytical methods to be thoroughly characterized regarding the stability, dissolution, formation of the protein corona, because this is then influencing the interaction with the cells and what the cell sees. And here we need other techniques to really evaluate what the fate of the nanoparticles is inside the cells. So we need a combination of very analytical techniques with different readouts. So we should not only use fluorescent readouts, but maybe also some other spectroscopic methods. And we just currently would like, or came up with this analytical platform, which we have now established with the knowledge in our institute. And we really focus on this characterization of nanoparticles in complex environment, which is in food products, cosmetic products, but also other products which are where we have nanomaterials, and I think these measurements are quite relevant also for the regulation. And we are also open for collaboration, and if you have questions, to use this service. So with that, I would like to thank the team, also especially to Alke for the great collaboration, all the foundations supporting the research, and you for your attention. Thank you.
Okay, thank you very much. I think we can take one quick question.
Could you just comment upon how you make the artificial fluid of lysosomes?
Oh, this is a recipe which is published. I can give you the composition. A lot of different ions. I don't know it by heart, but I can give you the composition. It's published, yeah.